is Joe McGirr, and I'd like to start off by passing around my study system so you can see it up close. And what you'll see are 3 species that comprise an adaptive radiation of Cyprinodon pupfishes. And similar to other adaptive radiations like East African cichlids and Darwin's finches, we see that these species have unique craniofacial morphologies that are adapted to different ecological niches. And today I want to explore genetic mechanisms that shape new traits like this, as well as help to maintain species boundaries. And specifically, I want to test predictions of ecological speciation theory that suggest divergent ecological selection leads to the evolution of new traits, and that genetically based reproductive isolation evolves as a byproduct of adaptation. And we can think about this as happening at the level of gene expression, where we have cis and trans regulatory elements binding to regulate some expression level of some gene. And if we have 2 populations that are diverging to different niches, divergent selection could favor the cis and trans regulatory elements to become co-adapted to cause low expression of a gene in one population and higher expression in the other. And if these divergent populations come together to form F1s, these co-adapted alleles can become decoupled, potentially leading to expression levels in F1s that are much lower or much higher than expression that we see in parental populations. And so this is my main question for today is, can divergent ecological selection cause hybrid gene misregulation? And if it can, we might expect to find genes that are important for divergent adaptive trait development to show differential expression between species and misregulation in their F1 hybrids. And this question has been asked before, but I think remains unanswered largely. A lot of what we do know about hybrid gene misregulation comes from crosses between distantly related species and subspecies of Drosophila and mouse. And in these crosses, misregulation is often associated with strong intrinsic reproductive isolating barriers like hybrid sterility and inviability. And misregulation is thought to result largely as a process of stabilizing selection rather than divergent ecological selection. And that's why I'm interested in asking these questions in this adaptive radiation of pupfishes. So for the remainder of my talk, I'll introduce you to this system. I'll describe those 2 different expression patterns that I've predicted to find differential expression between species and misregulation in hybrids. And then I'll discuss some exciting results that suggest that ecological divergence can potentially lead to hybrid gene misregulation. So why study this question in pupfishes? Well, they live on some really pretty islands in the Caribbean. My lab and I would go down and search for species like this, Cyprinodon variegatus. This species has a really wide range from the coast of the United States down through the Caribbean to the tip of South America. And throughout this range, pupfishes are largely dietary generalists, meaning that they consume mainly macroalgae and detritus. However, there are 2 really interesting exceptions to this rule on the small island of San Salvador, where there's been a recent adaptive radiation. And we find our specialist pupfish, our generalist pupfish, along with 2 endemic specialist pupfishes. One of those specialists, Cyprinodon bronitoroides, has evolved these short, robust jaws, as well as this nasal protrusion here that it uses to crush hard-shell prey like mollusks, and so I'll refer to this as the molluscivore. And then the other specialist, Cyprinodon disquamiter, has evolved these really long jaws, as well as this more elongated body that allow it to bite scales off of the sides of other fishes. So I'll call this the scale eater. And what's really great about this system is that this radiation is very young, around 10,000 years old. So these species are young enough that they readily hybridize in the lab. But despite their young age, we see drastic differences in adaptive craniofacial morphology. And I've done some work in the past looking for genetic variation associated with divergence in those traits. And I found that there are surprisingly few fixed SNPs distinguishing species. So we see the most fixed SNPs between specialists, fewer between generalists and scale eaters, and less than 100 SNPs out of 14 million fixed between generalists and molluscivores. And so the reason I want to ask this question about gene misregulation in this system is because there's been recent ecological divergence in sympatry that's led to the evolution of these species that look and behave very differently but show low genetic divergence. All right, so the first part of answering my question is to look for patterns of differential expression between species. And for this, I have 124 transcriptomes that I generated from crosses between populations from San Salvador as well as crosses between populations of generalist outgroups from New Providence Island and from North Carolina. Then I also have 58 resequenced genomes, so we can look at phylogenetic relationships between populations where our sympatric San Salvador species are more closely related, and then with increasing geographic distance between allopatric populations, you get increasing genetic divergence. All right, so to generate purebred F1 transcriptomes, I took breeding pairs, all from the same population, and collected their eggs, let them develop for 8 days, and then I extracted RNA from whole individual larvae to produce 3 to 5 libraries per cross. And then I asked, can I use these transcriptomes to find genes that are significantly differentially expressed between populations? And I used a standard pipeline in the program DESeq2. So for example, between the specialist populations, I found that there are over 4,000 genes significantly differentially expressed. And so that's the first part of the first pattern I expected to find. And then now to look for a gene misregulation, I again set up crosses, but this time between species, and then extracted RNA from their F1 hybrids with the same methods. And then I also sequenced their parents so that I could look at regulatory mechanisms underlying expression and divergence between species. And then I had reciprocal crosses for a lot of crosses to control for maternal effects. So here I wanted to take these F1 hybrid transcriptomes and ask, can I find genes that are significantly differentially expressed between F1 hybrids and purebred F1s generated from their parental populations? And I found about 1,500 genes were misregulated in hybrids that resulted from a specialist cross. And if we look at the total number of genes misregulated, that amounts to about 10%, which was pretty striking. And if we look at the crosses between generalists and each of the specialists, the other scale leader cross, their hybrids had similarly high misregulation, and then very little misregulation in the generalist by Molescovore cross. And then we can look at the crosses between San Salvador generalists and those outgroup generalists, and we see that the cross with New Providence Island, there was, again, very low misregulation, but then pretty substantial misregulation with the North Carolina cross. And a point I want to make here is that if we use branch lengths from that tree I showed you and do a phylogenetic least squares, it doesn't look like hybrid gene misregulation is explained by geographic distance between populations. So in other words, this misregulation here is much higher than we would expect given phylogenetic relationships between these sympatric populations. Okay, so now we have both pieces of the puzzle. We have differential expression and gene misregulation. And excitingly, we do see a lot of genes that are differentially expressed between species and misregulated in their F1 hybrids, and not just in the specialist comparisons that I'm showing here where 300 genes overlapped. I also found overlap in the crosses involving generalists and each of the specialists, so that in total there were 750 genes that showed this expression pattern. And so my next question was, do any of these genes show signs of selection? And so as a first pass, I just asked, are these genes near variants that are fixed between populations? And also, do they show evidence of high between population divergence measured as DXY? And I found that, yes, there were 125 genes that matched this criteria, and I'm referring to them as ecological DMI candidate genes. DMI because they show patterns consistent with divergent regulatory alleles negatively interacting in hybrids to contribute to misregulation, and ecological because these genes were strikingly enriched for biological processes related to adaptive trait development. So we see enrichment related to craniofacial development, skeletal muscle development. Here I'm showing that specialists tend to have larger adductor muscles, which is important for the force of jaw closure. We see enrichment related to nitrogen metabolism. We know that the specialists occupy a higher trophic position and have more nitrogen-rich diets than the generalists. And then we also see enrichment for a lot of terms related to brain development, which could affect a number of different behavioral differences between species. But we can also go beyond gene ontology enrichment to find convincing evidence that some of these genes are influencing specialist traits. So for example, MPP1 is differentially expressed between generalists and scale leaders and shows misregulation in their F1s, showing lower expression. Mutations in this gene are known to cause craniofacial deformities in mice and humans, and knockouts in mice are shown to cause decreased bone density. this gene shows differential expression between generalists and scale eaters, as I said, but they also show expression patterns that are consistent with being differentially expressed due to cis-regulatory divergence. This gene shows allele-specific expression in hybrids where the scale eater alleles are more highly expressed than the generalist alleles. And in short, this is really cool because it suggests that there's some linked variation nearby MPP1 that's controlling expression divergence. And we find 170 SNPs that are fixed between populations. That's within 20 kilobases of the gene. The gene region is in blue here. The region also shows signs of selection with high between populations divergence and low within population diversity. It shows a shift in the site frequency spectrum consistent with a hard selective sweep in scale eaters, as shown by negative Tajima's D, as well as composite likelihood ratios calculated by the sweep finder program sweep. And then finally, this region was significantly associated with jaw size using a genome-wide association mapping approach implemented with the software package Gemma. So together, this type of evidence for this gene and other candidates like it suggests that these genes are being differentially expressed between species due to their effects on adaptive trait development. But what does that mean for misregulation in the hybrid for important genes like these? And I told you that misregulation in other systems is associated with strong intrinsic barriers like hybrid sterility and inviability, which we don't see between these species. So perhaps misregulation is contributing to weaker intrinsic isolating barriers or even extrinsic isolating barriers where misregulation leads to abnormal development of anatomical and metabolic phenotypes that make hybrids perform poorly in the wild, which is supported by work that shows that hybrids with intermediate craniofacial phenotypes do very poorly in the wild, and also feeding kinematic data that shows that F1 hybrids differ in traits from their scale eater parents that are important for scale eating, like peak gape of the jaws and gape angle of the jaws. And then finally, I've looked at expression in older hybrids that are about 20 days old, but this time I specifically dissected craniofacial tissues that are divergent between species. And I found that misexpression is even more extensive in these adaptive tissues, even more so than what I've shown you in the 8-day-old bull larvae. And so what I've shown you is that misregulated genes under selection are associated with adaptive ecological traits in specialists, but I'm hoping that one of the broader takeaways is that this data supports a compelling genetic mechanism of rapid speciation where these ecological DMI candidate genes are simultaneously influencing adaptive trait divergence and contributing to reduced hybrid fitness, potentially leading to reproductive isolation between these young species. And with that, I'll thank my lab, my advisor Chris Martin, and my funding. And if you enjoy hearing about pupfish genomes, my lab mate Emily has 250 pupfish genomes, so check that out.